Hello, welcome back to ID Rapid Reviews. Today we review a 2026 study by Karanika and colleagues in the Journal of Clinical Investigation describing an intranasal dendritic cell-targeted DNA vaccine targeting drug-tolerant Mycobacterium tuberculosis persisters. Tuberculosis remains a leading cause of infectious mortality, and current regimens require prolonged multi-drug therapy. This is partly due to drug-tolerant persister populations. These persisters, driven by the stringent response pathway, enable metabolic dormancy and contribute to relapse. This has led to global prioritization of therapeutic vaccines to complement antibiotics. The authors developed an intranasal DNA vaccine. This intranasal DNA vaccine encoded REL-MTB, a key persistence phase antigen, fused to the dendritic cell-homing chemokine MIP-3 alpha. This design targets antigen delivery to dendritic cells, enhancing T-cell-mediated immunity against persisters. Preclinical efficacy was evaluated in murine tuberculosis models. Mice received either standard first-line therapy or a drug-resistant tuberculosis drug regimen, with and without the intranasal MIP-3 alpha REL-MTB DNA vaccine. Note that formal relapse assessment was performed in C57 Black 6 mice. C3HEB FEJ mice were used primarily for pathology evaluation. Outcomes included longitudinal lung colony-forming units, relapse after treatment cessation, and lung histopathology. Stable cure was defined as culture negativity with no relapse after stopping therapy. Immune mechanisms were assessed by measuring REL-MTB stimulated T-cell responses using flow cytometry. Additionally, dendritic cell recruitment and activation was also assessed. Spatial assessment of dendritic cell T-cell interactions using immunofluorescence-based spatial analysis was also performed. Translational relevance was assessed in non-human primates using blood and BAL samples. When added to first-line therapy, the vaccine significantly reduced lung bacterial burden by up to 3.6 log in C57 Black 6 mice at 6 weeks, and improved lung pathology in C3HEB FEJ mice. Importantly, this effect was antigen-specific, as no benefit was observed with the MIP-3 alpha ESAT-6 platform control vaccine. The vaccine accelerated sterilization, achieving culture negativity by 12 weeks in all vaccinated animals. Critically, 12 weeks after stopping treatment, none of the vaccinated animals relapsed, whereas all control animals receiving RHZE alone had detectable bacteria, establishing a stable cure phenotype. Mechanistically, the vaccine generated robust antigen-specific T-cell responses, interferon gamma, TNF-alpha, and IL-17A, and increased dendritic cell recruitment and activation. It also enhanced spatial proximity between dendritic cells and T-cells, suggesting improved innate adaptive immune synergy. These findings represent correlates that may contribute to improved bacterial clearance. Importantly, REL-MTB stimulated T-cell responses producing interferon gamma, TNF-alpha, and IL-17A were observed in non-human primates, both in peripheral blood and BAL, mirroring murine immunogenicity and supporting translational potential. These responses were sustained through 24 weeks. However, no efficacy challenge study was performed in macaques. These findings are limited to preclinical models. Human safety, immunogenicity, and treatment-shortening effects remain unknown. Also, MIP-3 alpha ESAT-6 served as a platform control to isolate the antigen variable, rather than as a benchmark therapeutic vaccine comparator. Additionally, optimal antigen selection requires further study, as other latency and persistence antigens were not directly compared. Finally, the C3HEB FEJ model assessed pathology, but did not include formal relapse testing. Therapeutic vaccination with the intranasal DNA vaccine accelerated attainment of stable cure and reduced relapse in preclinical murine models when used as an adjunct to chemotherapy. The induction of concordant REL-MTB-stimulated T-cell responses in non-human primates supports translational potential and provides a framework for defining immune correlates of protection for future TB vaccine trials. Human evaluation is the next critical step. Thank you for watching ID Rapid Reviews. Read the full paper in JCI.